Hey class, how's it going? I wanted to give this video lecture thing here a try. I'll be posting an Edpuzzle for you to watch, and you can use it for some other assignments and look back at it as you need for studying. But here we go. We're talking about cell division. So we're looking at how do cells replicate themselves. And I guess before we get started, if you don't have a piece of paper out in front of you to take notes as we go through this, I would pause the video now, get that paper out, and have something to write with. All right, here we go. All right, so you've seen something similar to this circle over here on the right. This is the cell cycle we talked about, or you learned about the other day, with the G1, the S phase, the G2 phase, and then the M phase here on the left. So cells have two major periods in their life. They have interphase is the main part of their life. That's the G1, S, and G2 phases all in one, right? Those are all interphase. You'll see in the middle here, there's the purple. It shows that all of those are interphase, okay? And then you have the M phase. This is the part of a cell's life where it actually splits into two cells, okay? And the interphase part is a couple things. It's the cell is doing its normal function, right? It's whatever the cell's meant to do, it's doing it. That's just living its life, okay, in interphase. And then if it's a cell that's going to divide, when it's ready, it will go into the S and G2 phases from there before it goes into the M phase, which is the actual splitting. That's the replicating itself of the cell. So the function of the M phase is to produce more cells for like growth or healing. Right? All right, so DNA replication. This is something where this is the DNA, like the cell making a copy of a DNA inside so that there's two copies of the same thing. So when it splits, each new cell can have its own copy. This happens during the S phase of interphase. We'll get into a little more detail on that in a second. Mitosis, there's 4 steps in mitosis. There's an acronym we use, which is PMAT, P-M-A-T. That'll help you remember the order of these 4 steps. So feel free to, I would write that down, P-M-A-T, and then as we're going through it, you'll realize what the acronym stands for. This is the division of the nucleus itself. Okay, so the nucleus has the DNA in it. It has to split, separate, and then new nuclei form so that you can have 2 different cells, and that process is called mitosis. Okay, so that's not the entire division of the cell. Okay, the finishing product, that what actually causes the cell to divide into 2, is called cytokinesis. That's the actual division of the cytoplasm. So once the nucleus is divided and all the organelles have split and separated, then the cytoplasm can split. It starts happening near the end of mitosis. We'll see as we get further down what I mean by that. And this results in 2 new cells. We call them daughter cells. So a couple vocab things to look at here, which it can be a little bit confusing. So I'll move my camera here in a second. But on the left, we have the DNA. When it's loose, it kind of looks like spaghetti. In the nucleus, this is when the cell is just doing its normal function. We call it chromatin. Okay, so chromatin is when it's loose. All right. Before it can split, after it's replicated, before the cell can split, the DNA condenses into these tightly packed things of DNA. Okay, the tightly packed things of DNA are called chromosomes. Okay, and remember, there's 2 copies of each chromosome, right? So when you'll see these are kind of X's, those are the 2 copies stuck together. So they're the exact same thing stuck together in the middle there. Each one of those 2 is called a chromatid. Okay, a chromatid, or sister chromatids. And then at the end over here, see they have to split because we're going to form 2 new cells. So once they split, now each one of those chromatids, it's still called a chromatid, but they're their own chromosomes now because they're not connected anymore, which is a little confusing. So it goes from 4 chromosomes, because they're all connected, and 8 chromatids, to 8 chromosomes, because they've split, and still 8 chromatids. So a little confusing, but that is what it is. So I just wanted you to see, don't worry about all these labels, just the difference between the different DNA structures. So on the left, we have our chromatin. That's like a bowl of spaghetti, a loose DNA there in this nucleus here. And the nucleus is cut in half so you can see inside. Okay, then you see, this is like the DNA has like it's surrounding by all these proteins and stuff. This is starting to condense, where it condenses to form this chromosome, right? Two chromatids connected. So each one of these two is an exact copy of each other there in that kind of X-shaped thing. All right, so you should be able to kind of try and describe this in your own words, maybe on your notes there. You can pause it to work to describe that. I'll go back to the previous slide here, so you can pause it there if you'd like. Here it is, pause if you'd like to copy down your understanding. All right, so when we were looking at chromosomes in science, we organize them like this, right? This is humans right here. We have 23 different chromosomes, but we have 2 copies of each because you have one from mom, one from dad, right? So you have a total of 46 chromosomes, but it's 23 duplicates of each other, except for at the bottom here. If you're a male, you have the X and the Y. If you're female, you would have both X and X chromosomes here. This is just something just to see how we depict it as a scientist. All right, so interphase, not part of mitosis, okay? Still part of cell division, but not part of mitosis, okay? This is those 3 different phases, G1, S, and G2. The cell is performing its normal functions. This is just the cell living its life. If it's going to divide, this is where it will start to prepare for cell division before it goes into mitosis. Interphase, the first phase of it is G1. This is all really that's happening here is the cell is growing a little bit, okay? Because it was just split, so it has to grow a little bit to become its normal size. The DNA is still in chromatin. It hasn't condensed. It doesn't condense until we get to mitosis. Okay. Now, the S phase, this is when DNA replicates itself. So S stands for synthesis. We've talked about that, meaning to build. So it's building a copy of itself here. So it's duplicated, it's replicated, still in chromatin, still spaghetti-like. There's this structure here called a centrosome that you need to know. It kind of looks like the T here. It makes a copy of that as well. That's going to come in handy when we get to mitosis. Okay, that's something that is used to pull these chromosomes apart when they turn into chromosomes. Okay, all right. DNA replication, the S phase. Okay, this is what I just talked about, how the DNA replicates itself. So we know DNA is this kind of double helix, this spiral shape. You'll see on the image on the right here, that spiral comes apart and splits in 2. Okay, so now it's each half of that DNA, and then a new half gets added to each side of that. As you see here, there'll be an animation that you can look at in a second that really depicts it well. And the end result is 2 copies of the same exact piece of DNA. Each copy is half old, half new, right? So you see here as it splits, you'll see how there's the old piece and the new piece being added to each side. And this animation shows it well. Always feel free to pause if you need to finish copying down. This is an animation here showing, right, on the right, we have our DNA. It splits, right? One strand goes up, one strand comes down, and a new half gets added to each one of those. So it's a pretty cool process, pretty amazing that our bodies can do this. I'll leave it here for a second just so you can observe it. And there's these different structures that we'll learn more about in a little bit. But these are proteins that are facilitating this, right? So we've talked about how important proteins are. One thing that there's some proteins do is work to replicate DNA. All right, so interphase, the cell grows, right? Feel free to kind of pause it here. Use this for the other assignment that we're going to be talking about, which is you're going to be drawing and describing each of these processes. So cell grows, again, in preparation during the G2 phase. This is the final part of interphase. Okay, one more time it grows. Now it's getting even bigger because it's going to split in half. All right, now we're moving into mitosis, right? We're done with interphase. We're moving into, all right, now the cell is trying to split. So mitosis is 4 different parts. We said it was PMAT, right? P-M-A-T. The P stands for prophase. Okay, in this phase, what's happening is the nucleus breaks down, so the nucleus dissolves. Okay, the chromosomes, or the chromatin, condenses into chromosomes. So see that in the animation up here. Move this to the side here. You see that in the animation. The next slide has a larger version of this animation, but the chromosomes are condensing. They're forming by the condensing chromatin. It's already been replicated, so no more replication. It's just there's 2 copies there already, and those chromosomes, right? The 2 chromatids, the 2 different exact copies of each other, are held together in the middle by something called a centromere. Okay, that's the structure in the middle, and that'll come into play later down the road. Okay, the centrosomes, those T-shaped things, they migrate to opposite sides because remember they're going to be used to pull the DNA apart. So they migrate to the opposite sides. These fibers that they produce to kind of grab onto and pull are called spindle fibers. They're starting to form in this phase, but you kind of see them right there start to form, to pop out. All right, I'm going to go to the next slide. That is a larger animation. So see, they just condensed the chromatin condensed in the chromosomes here. You see some spindle fibers start to form out here on the sides. Okay, so you can pause it here, use this animation, or go back a little bit to draw your prophase and give your explanation for the assignment there. All right, so if we were in class, we'd be doing a paired deck here where we could do this. All right, then we have the M of PMAT, which is metaphase. This one's relatively short, so you'll notice that these chromosomes, they've already condensed, they start to line up in the middle of the cell, okay? And then on the sides, the centrosomes are in the middle of the chromosomes. The centromeres are putting these spindle fibers out that are going to connect to that center piece. So they're lining up, spindle fibers form from the centrosomes and are attaching to the centromere in the middle of the chromosome. Okay, again, you can pause here, do your metaphase one, do your explanation. This is a larger animation here. All right, so then we have anaphase, the A in PMAT. The centromeres split, so the center piece of the chromosomes, it splits. So now these chromosomes can be pulled apart, right? So the chromatids, each half of that chromosome, they're exact copies of each other, are pulled apart by the spindle fibers. Okay, the cell is starting to elongate, so you see it's kind of turning oval shaped because it's going to split down that middle. So this is where cytokinesis starts to happen. That elongating is where cytokinesis happens, which remember is different than mitosis. Mitosis is specifically about the nucleus. This ends when the chromosomes stop moving when they're on either side. Okay, so you can pause it here if you need to work on that assignment part, do the anaphase portion. See them splitting apart, and they will pause eventually. Once they pause, anaphase is over, and we will move on to the final stage, which is telophase. Okay, the new nuclei, plural for nucleus, they begin forming around those chromosomes. Chromosomes begin to unwind back into chromatin, turning back into that spaghetti shape. The parent cell starts to pinch in the middle. Okay, it's not a part of mitosis. That's cytokinesis happening, but you see it starting to pinch. So this is where cytokinesis and mitosis overlap as well. Pause if you'd like to use this for your assignment. I got about 2 minutes left, so I'm going to kind of hurry through this before it doesn't let me record anymore. All right, we can see how the nuclei form and it starts to pinch. Okay, that is telophase. Okay, then finally we have cytokinesis. Right, this is the final split of the cytoplasm, different than mitosis, but still a part of cell division. Okay, it starts in the late anaphase, so they're overlapping, remember, and it finishes at the end of telophase. Okay, these microfilaments, these little fibers, kind of like they're part of the cytoskeleton, they start to pinch. Okay, and that forms what we call cleavage furrow. So that's the indent there, down where it's going to eventually split. It forms two daughter cells. We had a parent cell, now two daughter cells. That's what they call them. Okay, so you can pause it here to kind of draw. Now these are basically the same drawings that you would have done for interphase, right? But there's two of them, okay, because these are just two new cells now after cytokinesis is done. They're a little smaller. They'll grow a little bit, but this is that. Feel free to pause if you need to. Here's our cytokinesis. Awesome. Yeah, we don't need to go there. All right, so thanks. Feel free to watch it multiple times. Feel free to pause, go back, whatever you need to do to finish out your notes and be able to complete the assignment here. I'll show you where you're going to be drawing each of these steps, labeling what stage it is, finally doing a summary there at the bottom. On the top, we have a word bank here to use for your labeling. Some of these words will be used more than once because they're within different portions of the stages of the cell division. All right, guys, I hope you're doing well. Miss you guys. I will talk to you soon.